Hello, good evening, very good evening. Very few participants had joined so far. Uh, Maybe they join in later part because today was an important day actually. Um, Maybe it's because of the Sunday thingy, because it's weekend. People join very less, or because I was writing this on continuous basis that we will have a class about protein isolation, so be there on time. Anyhow, we will wait till 16:05 to let the crowd join, and then we will start. Today we're gonna discuss about protein isolation, quantification and purification. And how are you all? How is the weather today in Delhi? We had a lot of rain. Makes the weather very good and yeah, very relaxed. I had a little bit nap in the afternoon. I just woke up at 4:45. That students were waiting for the class, and just in my dreams, and I just woke up. Oh, I have a class actually. So I just started. Okay, crowd is coming now slowly and slowly. That's good. Okay. this thing does not work. So we can start now, I guess. So basically in this we will deal with how we homogenize our sample, how we fractionate it, how we centrifuge, how we quantitate, chromatography and electrophoresis. So all these steps are taken care in that part. So these are famous people who got the Nobel Prize winners on chromatography and electrophoresis. So Orchard John Porter Martin in 1952, he got in National Institute for Medical Research, London, United Kingdom, for the invention of partition chromatography. Richard Lawrence Millington Synge, 1952, he got for the invention of partition chromatography. Then Arne William Torren Tisles, 1948, he got for the electrophoresis and adsorption analysis. Then John B. Fn, 2002, for the development for identification and structural analysis of biological micromolecules. And Koichi Tanaka for the development of soft desorption ionization method for mass spectroanalysis of biological micromolecules. And Theodor Svedberg for his work on the disperse system. And in organelle fractionation, to separate your various internal organelle of your cells. So Albert Collaudet, Christine Duvo, George Pillade, so from 1899 to 1983, 1917 and 1912, they all got the Nobel Prize winner in their respective years for organelle fractionation. Then Frederick Sanger and Frederick Sanger in 1958 and 1980, they got the University of Cambridge, from the University of Cambridge, for his work on structure of proteins, especially on that of insulin. Then Frederick Sanger in 1980 for MRC Laboratory of Molecular Biology, for the contribution concerning the determination of base sequence in the nucleic acid. Then comes your protein isolation. So homogenization is first step in the protein isolation, that is disrupting your cells, breaking out the cell membrane. Then the fractionation, crude separation of homogenates into the fractionation. So homogenization step, you have Downs homogenizer with sonation, osmotic shock and French press. Then fractionations, fractional precipitations, ammonium sulfate precipitations and differential centrifugation. So you have homogenate forms, they get centrifuge at 500 G for 10 minutes, that is a supernatant you get. Then, but the pellet will have your nuclear fractionation after the centrifugation. Then at 10,000 G for 20 minutes, you will get the pellet is mitochondrial fractionation. Then at the bottom, you will get your microsomal fractionation. Yeah. Then comes your protein isolation. This is the centrifugation to separate particles and macromolecules based on the mass, density and shape. So you have your differential centrifugation, that is, sample is poured into the tube, you have more dense particles and less dense particles, and then you centrifuge with the centrifugal force and particles settle according to the mass. Then you have a pellet, and then the pellet you have to decant your container, and then yeah, you can get your things done. And in the rate zonal centrifugation, you have large particles, smaller particles, you mix them together over the sucrose gradient. Then you centrifuge them. After the centrifugation, your particles get separated according to the density, and then you can separate them according to the mass of particle. Yeah, first we did the fractionation, then we did the first we did the homogenization, then we did the fractionation, then we did the centrifugation in order to isolate your specific type of organelle that you are interested in. So at different G and for different minute, you can separate your particles. So like 300 G for 5 minutes, you can get large organelles, and then 10,000 G for 10 minutes, you have mitochondria, inclusion bodies. Then 40,000 G for 30 minutes, you have membranes, plasma membrane, tonoplast, microsomes and so on. At 200,000 G for 16 hours, large protein complexes are coming out. Then at 400,000 G for 24 hours, you have proteins like 50,000 Dalton. Yeah. That's how they are all separated. So this is the sedimentation velocity technique. We can see you have a low density solution and high density solution. You mix them together with layering of the samples on the top, and then separation by sedimentation coefficient, then infiltration collected through hole in the bottom of the tube later on. These are the various sedimentation velocity technique, the densities like RNA density, DNA, most virus, and the sedimentation coefficient. You can separate all these molecules according to these values. Then comes, after the isolation is done, quantitation, that how much is a protein is present in your sample. The first method is the Lowry method, in which you can determine the protein from 10 microgram to 1000 microgram per ml. Yeah. So this was found by the Lowry. It was published in a Journal of Biological Chemistry in 1951. Then comes the Bradford protein determination. This technique is simpler, faster than the Lowry method, is subject to less interference. Yeah. So this Comasi blue G250 dye appears to bind most readily to arginyl lysyl residues of proteins. So this specificity can lead to variation in the response of the assay to different proteins, which is the main disadvantage of the method. So let's check how the Bradford assay in reality.
In this video, you will learn how to perform protein quantitation using the Bradford assay. Protein quantitation is one of the most commonly performed procedures in a biotechnology laboratory. In this activity, the protein concentration in milk will be measured using the Bradford assay. For this activity, you will need 1x Bradford reagent and 1x phosphate buffered saline, or PBS. Using a 20 to 200 microliter micropipette, set the pipette to 98 microliters. Pipette 98 microliters of 1x PBS into an empty microcentrifuge tube labeled sample. Switch to a 2 to 20 microliter micropipette and set the pipette to 2 microliters. Pipette 2 microliters of milk sample into the microtube labeled Sample A. Mix the milk sample and PBS by either pipetting or vortexing. This mixture has diluted the milk to 1 part in 50. Label 2 cuvettes, one control and the other sample. Do not handle the cuvette where the light passes through, and make certain your labels are well above this area. Pipette 20 microliters of diluted milk sample into the cuvette labeled sample, and 20 microliters of 1x PBS into the cuvette labeled control. Next, we will set up protein standards. See your protocol for the correct concentrations. Label a cuvette for each standard and mark it with the corresponding concentration. Next, add 20 microliters of each protein standard into the corresponding cuvette. Make certain you use a clean pipette tip for each sample. Now you are ready to add the Bradford reagent to all of the cuvettes you have prepared, including the sample and control. Add 1 milliliter of 1X Bradford reagent to each of the cuvettes. Mix completely by pipetting up and down with the micropipette. Incubate the cuvettes at room temperature for 5 minutes. After 5 minutes, visually compare the cuvette containing the milk sample to the cuvettes containing the protein standards. Determine the standard that most closely matches the color of the milk sample. Estimate the protein concentration of the milk sample based upon the visual comparison. To determine the protein concentration using a spectrophotometer, select for the Bradford assay. When asked to insert blank into the spectrophotometer, insert the cuvette labeled control. Use the blank to set the spectrophotometer to 0 absorbance, or 100% transmittance. If instructed, read the absorbance of the 7 protein standards and record the absorbance values. Remove the cuvette labeled blank and insert the milk sample into the spectrophotometer. Read the absorbance and record the value.
Yeah, so that's how we do the Bradford assay in practical, from practical aspect. Now there's also a way to quantitate your protein by ultrasensitive fluorescence protein based quantitation, in which you add your dye with hard sample, heat your sample for 10 minutes, then cool to the room temperature, then you can read the fluorescence. So you get the fluorescence versus protein graph. How much fluorescence you got, you can write the protein concentration in that. Then we are done with the isolation part. Now comes the quantitation part. Now comes the purification part. Yeah. So this is a dialysis which is used to remove small molecules from the protein solution or to change the buffer composition of the protein solutions. So dialysing tubing or bag is made of cellulose and has different cutoff range. So you have a dialysis bag here, yeah, and you have a concentrated solution bag, and you have buffer outside. And then, so what will happen? The small molecules that you can see here, they go out outside. Mainly the large molecules remain inside the bag. So in this way, by this dialysis, you can separate between small and large molecules of proteins. So let's check how this technique is done in practice. So there's another technique called chromatography, which is mainly, it's a term which refers to all chromatographic methods in which the mobile phase is liquid and the stationary phase may be liquid or solid. The first one in the chromatography is a gel filtration chromatography, or gel excision chromatography is also called. So in this we have a carbohydrate polymer beads in which your small molecules enter the aqueous spaces within the beads, so they get stuck there, where the green beads they will go flow out, and the large beads with the largest which cannot enter into the beads will also flow away. So in this way, when you enter into your sample, so according to molecular exclusion gel, they will start to get separated with red, green and yellow. So the yellow will separate first, then green and then red. Same thing has been shown here. So this is Cefadex G75 gel and this is Cefadex G10 gel. And this is your proteins that you want to get separated. So let's check how does we do this technique. That is your gel filtration Cefadex G50.
We're starting with gel filtration medium. This is Cefadex G50. The number here gives you an approximation of the size exclusion. So this is will exclude proteins about 50 kilodaltons, 50,000 molecular weight. Inside is a white powder. In this beaker I put 9 grams of the G50 powder, and I swelled the powder overnight using this tris buffer, 30 millimolar tris buffer, pH 8.0. You can see that the 9 grams has swollen up to a volume of about 90 milliliters, almost 10 milliliters per gram of solid. Now I'm going to gently stir the gel. It's important not to stir this too vigorously. Don't mechanically stir this. That can break your gel particles up into very fine dust, which will impede your flow rate in the column. So this is making a slurry. I'm using a sawed-off pipette to transfer my slurry to the column. For gel filtration, I want a fairly tall gel bed. So I'm trying to mostly fill this column. And excess buffer will drip out the bottom. So here you can see the drips coming out the bottom. Gel bed is starting to settle a little bit. Gel bed is right here. I'll add a little more buffer so that it doesn't run dry at the top. So I'm gently adding a little buffer to the top. This looks great. You can see the gel bed is fully settled here. There's a little bit of buffer on the top. I've capped the bottom right now to keep it from going dry. I'm going to put a mixture of 2 molecules onto the column. Blue dextran is a very large molecule. It's a polysaccharide. Molecular weight about 1 million. Fluorescein, on the other hand, is a small molecule. Molecular weight about 350 grams per mole. I chose these 2 molecules because they're nicely colored, so I'll be easily able to see the separation visually as they go through the gel filtration. I've drained the buffer, so now there's no residual buffer on top of the gel, and I've made a mixture of a total of 0.8 milliliters, 400 microliters of the fluorescein, 400 microliters of the blue dextran. Nice green color. I'll separate these on my gel. For this size column, this is getting near the maximum I would put on the column. I'm using a Pasteur pipette to gently load my material onto the column without disturbing the gel bed. So here you can see the blue molecules, the large molecules, are moving faster through the gel. So I'll hope that I can get a complete separation between the blue and the yellow. Now the excess on the top of the gel is all soaked in. So I'm adding buffer. very carefully to the top of my gel filtration column. I'll just add a fraction of a milliliter first to help get everything into the gel before I add a large volume of buffer. So now the separation has proceeded somewhat. I'm going to try to add a decently large volume this time of buffer on the top. About a milliliter would be great. If I do this well, then the buffer that I put on the top remains colorless because all the colored molecules have already gone into the gel. Alright, now you can start to see a little colorless layer on the top. That's perfect. All the colored molecules are in the G50 and they're starting to separate quite nicely. The yellow molecules are up here, the blue are down here. Still got a little overlap, so I may have overloaded this column, but we'll see. I'm hoping that before the blue molecules elute that they'll actually be separated from the yellow, but it's going to be close. So out of the bottom now, blue liquid is dripping. You can see I do have a colorless layer starting to develop here. The fluorescein has been slowed down by the gel filtration. The blue dextran is coming through. So this technique is often used to separate a protein, which is large, from the small molecules that could be salts or could be a fluorescent tag that is unwanted. I've topped up with buffer on the top so that it never runs dry. The fluorescein is now eluting from the column. Fluorescein is fluorescent. I'm shining a little blue light, UV light on it, and you can see it glows green. The last little bits of fluorescein are emerging from the column now. Here's the fractionation. This first tube has nothing in it. So this volume, which looks to me about 2.5 milliliters, is called a void volume, where nothing comes out. But then the blue molecules, the blue dextran, came through the column swiftly. So this is about a milliliter. Some of the blue dextran tailed into the next fraction. This is low concentration. Then I have a nearly colorless fraction here, and then the fluorescein starts coming out here. Fluorescein stopped coming out right about here. So the total volume will be 2.5, 3.5, 4.5, 5, 6, 7, 8, 9, about 10 milliliters is the total volume, which is what I would expect for a column of this size. Often what you want is just this first fraction with the large molecules. If you're trying to desalt a protein or remove the unbound label from a protein, then this is the fraction that has value. These fractions would just be junk. The original mixture, you can see, glows nicely, and what I believe is isolated blue dextran from the column has no glow in the UV. That's a sign that I have successfully separated out all the fluorescein from the blue dextran. So the column, if I've done it well, comes out completely colorless. It's reusable. Sometimes microorganisms like to grow in the Cephadex, so if you want to preserve your column for a long period, keep it in the fridge, certainly. And sometimes azide is added to the buffer to prevent growth of microorganisms. Here's tris buffer with 0.01% sodium azide, and this will prevent microorganisms from chewing on the dextran that's in the column if you're storing it over a long period of time.
Okay. Now this is a chromatography, ion exchange chromatography, in which basically you check, you differentiate according to their ion current, what kind of ion they have. So they are positively charged protein binds to the negatively charged beads here. So they have negatively charged beads. They will attach to this, these positive ions being attached to it, and you will have these negatively charged beads will attach to it. So you can flow it afterwards. So your negative will flow straightforwardly. Your blue one will remain attached to it. So protein in a solution that has low ionic strength is added to the column, and the protein interacts with the beads via electrostatic interaction. The resins carrying a positive charge are called an ion exchanger because the ions will exchange on this type of column. So resins carrying negative charge are called cation exchanger because cations will exchange on this type of column. Then comes your carboxymethyl group CM, this one, and this one is D-ethyl aminoethyl group. So in an ion exchange chromatography, the matrix carries a net positive charge which interacts with the negatively charged protein strongly. And in cation exchange chromatography, the matrix is negatively charged and interacts with the positively charged proteins. Let's see how is this done.
Ion exchange chromatography is a chromatography process that allows the separation of ions and polar molecules based on their charge. As well as other forms of chromatography, in ion chromatography, there is a mobile phase, which generally consists of buffer solution. And there is a stationary phase which consists of a matrix that contains charged ionizable functional groups. This type of chromatography is subdivided into an anion exchange chromatography and cation exchange chromatography. In anion exchange chromatography, negatively charged molecules are attracted to a positively charged solid support, which is usually beads that provide a positively charged surface, such as resin quaternary ammonium. In cation exchange chromatography, positively charged molecules are attracted to negatively charged solid support, which is commonly beads that provide negatively charged functional groups, such as resin methyl sulfate. Ion exchange chromatography is frequently used for the separation and purification of proteins. The basic process of this chromatography can be represented in 4 steps: conditioning or equilibration of the stationary phase, sample loading, washing, and elution of the retained molecules. The first step is the equilibration of the stationary phase to the desired start conditions. The pH and ionic strength of the sample buffer are selected to ensure that, when sample is loaded, proteins of interest bind to the medium, and as many impurities as possible do not bind. Therefore, the same buffer is used for the equilibration of the stationary phase. When equilibrium is reached, all stationary phase charged groups are bound with exchangeable counterions, such as chloride or sodium. When the column is equilibrated, the second step is sample application. Depending on the pH of their environment, proteins may carry a net positive charge, a net negative charge, or no charge. This net charge can be determined by comparing the pHi of each protein with the pH of the buffer. Proteins are large biomolecules, consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain specific to each amino acid. Depending on the pH of their environment, the amino group can be protonated and become positively charged. The carboxyl group can be deprotonated and become negatively charged, and the side chain of some amino acids can become positively or negatively charged due to the gain or loss of protons. So, the net charge on the molecule can be positive, neutral, or negative. The isoelectric point, or pHi, is the pH at which a protein carries neutral charge. At a pH below their isoelectric point, proteins carry a net positive charge. And at a pH above their isoelectric point, they carry a net negative charge. In this example, the pH of the buffer is equal 5, consequently, protein A has a net negative charge, and both protein B and C carry a net positive charge. In addition, protein C is more positively charged, since its pHi is higher than the pHi of protein B. If we apply cation exchange chromatography,
protein that has a net positive charge, it will bind to the negative charge beads of the stationary phase. On the other hand, uncharged proteins, or those with a net negative charge, would not bind. They pass through the column at the same speed as the flow of buffer. When all the sample has been loaded, the next step is column washing. The column is washed with start buffer. To ensure that all non-binding proteins have passed through the column. When all the sample has been loaded and the column washed, conditions are altered in order to elute the bound proteins. Most frequently, proteins are eluted by increasing the ionic strength of the buffer, or, occasionally, by changing the pH. As ionic strength increases, the salt ions compete with the bound components for charges on the surface of the medium, and one or more of the bound species begin to elute and move down the column. The proteins with the lowest net charge at the selected pH will be the first ones eluted from the column as ionic strength increases. Similarly, the proteins with the highest charge will be most strongly retained and will be eluted last. Protein B has the lowest net positive charge, so it can be eluted first. Then, protein C with the highest net positive charge, it can be eluted next, by increasing the salt concentration. Also, varying the pH of the buffer can be used to effect a separation, and to elute the retained proteins. As we've seen, the pHi of the protein B is equal 7, and the pHi of protein C is equal 9. So if we add a buffer with pH above the isoelectric point of protein B, and below the isoelectric point of protein C, the protein B will become negatively charged, and can then be eluted. The protein C is still charged positively, consequently, still retained. Next, protein C can be eluted if we apply a buffer with pH above its isoelectric point. So, it becomes negatively charged, and it will pass through the column. One of the primary advantages for the use of ion chromatography is only one interaction involved during the separation. Another advantage of ion exchange is the predictability of elution patterns. By controlling changes in ionic strength or in pH of the buffer, using different forms of gradient, proteins are eluted differently in a purified, concentrated form.
Then here comes affinity chromatography. So this was so far in detail about ion exchange chromatography, how that is done. Now comes to affinity chromatography. So this method relies on the knowledge of the properties of enzyme that you want to isolate. So basically in this, antibodies must be available that have been raised against the enzyme. Or enzyme must be known to be bind to some substrate such as a ligand non-covalently in a reversible way. So it is the most specific chromatographic method because it relies on the specific binding of the target proteins with its own specific ligand. So basically you have your proteins loaded into that with the red and blue. The red one will be recognized by the antibody, blue one's not. So the blue one will flow first, then the red one will flow later. So in all these chromatography techniques, one is separated because of the attachment to the beads that we had, and then rest is separated accordingly. So let's check how this is done.
In affinity chromatography depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and elution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. Non-binding molecules will pass through in the flow-through during washing with binding buffer. In the third step, elution, biomolecules are released from the biospecific ligand into the elution buffer by change in the buffer composition. A common way is to increase pH of the buffer.
Yeah. Now comes your main part. This is HPLC. This is high performance liquid chromatography. So in this high pressure liquid chromatography, which is an enhanced method, enhanced column method for protein separation, the resin size are most refined in them. So all chromatographic methods described above can be applied with HPLC columns and equipment with the HPLC. And the HPLC columns are packed with much smaller beads than those used in the standard columns. And this simple alteration greatly increases the number of theoretical plates. An intermediate version between column and HPLC is called FPLC, also fast performance liquid chromatography. Then the HPLC, the column is large tube containing the small particles known as stationary phase, and the chromatographic bed is composed by the gel beads alone when they are inside the column. And the sample is introduced into the injector and carried into the column by the flowing solvent. And once the column, the sample mixture separate as a result of different components adhering to the using diffusing into the gel. So as a solvent is forced into the chromatographic bed by the flow rate, the sample separates into various zones of samples, components, and these zones are referred to as bands. So here we see different bands 1, 2, 3, 4, 5. So just separating, being separated. So let's check how this is done.
HPLC stands for High Performance Liquid Chromatography, but could equally well stand for High Pressure Liquid Chromatography. It is used for separating mixtures, either to analyze the mixture or to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates. How fast each one moves depends on its relative affinity for the mobile and the stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase non-polar. In paper chromatography, the solvent moves along the paper by capillary action. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, Several samples can be run in succession by loading them into this auto sampler, which will run them in order without any human intervention. The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, a component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a nonpolar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
This is now how the chromatography mainly works. Now comes your next session of your FPLC, which is a fast performance liquid chromatography. It's a type of liquid chromatography where the solvent velocity is controlled by the pumps. So the pump controls the constant flow of solvents, and the solvents are assessed through tubing from an outside reservoir, and the flow rate of the solvent is set through the computer input and controlled by the pumps. So now comes your gel electrophoresis. We are done with so far the chromatography techniques, all the chromatographic techniques. Now it's the gel electrophoresis. So we will discuss mainly your 2D gel gross gel electrophoresis, how we do them. So protein separation, the most popular gel is your polyacrylate gel, so which forms a three-dimensional molecular sieve. And under denaturing condition, gel can resolve proteins of size from 10 to over 250 kilodalton. It is the most powerful one. So proteins and nucleic acids migrate in electric field according to the net charge, and for this reason separations can be achieved. So in many cases, electrophorese is coupled to a second separation method to achieve very high resolution of different proteins. The most popular method is SDS page, where electrophorese is coupled to a sieve device that differentially retards large proteins. So SDS page, the key feature is that the SDS coated proteins have the same electrophoretic mobility, and thus molecular sieving effect is the major parameter that causes separations of proteins. And the free flow electrophoresis, this is the simplest in concept but most difficult to achieve, requiring expensive equipment. Buffer of very low ionic strength is passed slowly down two large glass plates separated by a narrow gap. So protein mixture is injected at the top of the plates, begins to flow down with the buffer flow. And the buffer flow is adjusted with the protein never achieves the electrode. So here we can see you have your all green, yellow and red molecules, and they put over the gel, so they will separate according to the size. The smaller one will be at the bottom, green will be at the middle, and the yellow will be at the bottom or the top. So in this way you can separate your molecules. And also acrylonitrile and methyl bisacrylonitrile, they together make this chain of your network of pores through which your proteins will transfer. Then SDS is also added in the page in polyacrylate gel. We're also adding SDS. SDS will coat the proteins it has a negative charge, will go around it and let the separation of protein happen according to their size also. So in this way you can see various proteins are being separated according to the size. You can save it for data keeping later, and this is a different staining time, like for 1 hour de-staining time, and with different reagents you can see different results. Now based on these gel electrophoroses, there's a thing called isoelectric focusing gel electrophoroses. So in this case, first what we do is our protein molecules, they are multi-charge polymers, yeah, positive and negative. So we separate them according to the different high pH and low pH. One side we give high pH, on the other side we give a low pH. So they will find the gel contains the polyampholytes with form pH gradient in the gel under the electric field, and proteins can then separate it based on their pI values. So the pH gradient polyampholytes with many pI values differ by 0.01. So this is isoelectric focusing gel electrophoroses, unmodified, modified, unmodified and modified. Can detect difference of 1 charge per molecule. So let's see how protein precipitation for 2D gel electrophorese is done. So this is the protein precipitation, how protein is precipitated in order to prepare for your 2D gel. Yeah. So now after running your gel over the this pH strip, isoelectric focusing is done. So your proteins are separated according to the pH. Then you run over the 2D gel, that is run on the SDS electrophorces. Then your proteins will be separated on this gel. Then first you separate it according to the pH, then you separate according to the size. So this kind of pattern you will see. This is the whole proteome of one cell sample. So it will show all the proteins that are present in one gel. First based on the pH, then based on the size. Yeah. So these belong to one pH, these belong to one pH, these belong to one particular pH. These are positive pH, these are negative pH, and these are the smallest size and these are the largest proteins and so on. So now let's check how this is done in practically 2D gel electrophorces.
Hi, I'm Steve Freeby. I am a staff scientist at Biorad Labs. In this video, I am going to demonstrate how to run good, reproducible 2D gels. The video is going to be broken into 3 segments. The first segment will be how to apply, sample, and rehydrate the IPG strip. The second segment will be on how to equilibrate the IPG strips in preparation for running on the second dimension gel. And the third segment will be staining of the second dimension gel. In this video, I hope that you get tips and techniques and tricks that will help you run good quality 2-dimension gels. After sample preparation, the first step to 2D is to rehydrate the IPG strip. And I'm going to demonstrate this process using Biorad's 2D starter kit. The 2D starter kit is a great tool for learning 2D gel electrophoresis. It contains all the reagents and an E. coli sample for running a 2D gel. The first step is to prepare the sample and the reagents according to the instructions in the manual. The components of the rehydration buffer are urea, CHAPS, DTT, ampholyte, and bromophenol blue. I will be loading 200 micrograms of E. coli sample that has been reconstituted in rehydration buffer onto an 11 centimeter pH 4 to 7 IPG strip. To rehydrate the strip, I will load 200 microliters of sample into the focusing tray in each lane that I'll be running a strip. I am using a focusing tray that is specific for 11 centimeter strips. Trays are available for all IPG strip lengths. Tilt the tray toward you to allow for even distribution of the strip. An easy way to do this is to take the focusing tray, put it on the edge of the lid to the focusing tray, and you have the tray at an angle towards you. Dispense sample along one edge of the lane of the tray. And you want to dispense it in a nice even bead from one electrode across to the other. One even bead. I have shown you this with the focusing tray, but this can also be done with the rehydration tray. Now we are ready to place the IPG strip into the focusing tray. An IPG strip has 2 sides. One is a plastic protective backing and the other is a dried gel. You begin by removing the plastic protective backing with forceps, and you can tell where that plastic protective backing is by a slight indent on one side of the plastic pieces. The side that has indented is your protective backing. You pull that rapidly across, and now you have gel side up. Take your forceps, hold onto the strip. On the negative side, you'll see the other side has a positive side. The negative side is the opposite of that. And you can always tell when the IPG strip is facing down, but you will be able to read the numbers of the pH range of the IPG strip in the correct orientation. Now the gel will be loaded onto the sample bead, gel side down. If you are running multiple samples, I recommend labeling your strip before this step. Make sure to label the side of the gel and not the protective backing. To load the IPG strip into the focusing tray, place the positive end of the IPG strip into the positive electrode of the focusing tray and slowly drop your IPG strip into the lane of the focusing tray. Being very careful not to trap any air bubbles. If there are any air bubbles trapped, you can pick up the end and gently reposition the IPG strip until the air bubbles are expelled. If you don't remove air bubbles, there will be areas in the IPG strip that do not rehydrate properly, and that will cause serious anomalies in your first dimension isoelectric focusing run. The initial step of the first dimension separation is to rehydrate the strip. I will do passive rehydration in the focusing tray. Other forms of rehydration are active, which may be used for high molecular weight proteins, and cup loading, which is ideal for the separation of proteins that will be separated at extreme basic and acidic pH. Place the focusing tray into the protein IEF cell, making sure to align the positive electrode of the focusing tray with the positive electrode of the protein IEF cell, and add enough mineral oil to cover the strip in each lane. The purpose of the mineral oil is to prevent the strips from drying out during the course of the rehydration and the run. Now I am going to program the protein IEF cell. Instructions for programming are provided in the protein IEF cell manual. Typical rehydration times are 12 hours and focusing time ranges from 6 to 8 hours. At the completion of focusing, if you do not have time to run the second dimension gel, first remove the strips from the focusing tray, blot off the excess mineral oil, and place the strips into a rehydration tray at minus 80 degrees C. The steps following rehydration and focusing of the IPG strip are equilibration of the IPG strip in preparation for the second dimension gel, which I will now demonstrate. The equilibration process is a two-step procedure. Equilibration buffer number 1 contains DTT for reduction of proteins in the strip. Equilibration buffer number 2 contains iodoacetamide for alkylation of proteins in the strip. Each solution contains SDS for denaturation of proteins in the IPG strip to ensure optimal performance in the second dimension. Prior to equilibration, I will remove the IPG strip from the focusing tray and blot the excess oil off of the strip. First, pick up one end of the IPG strip, turn it over so the gel side is up, and place the IPG strip gel side up on a dry piece of filter paper. Wet another piece of filter paper and place the wet filter paper onto the IPG strip and gently blot the excess mineral oil off the strip. Be careful not to press too hard to damage the IPG strip gel. The next step is to equilibrate the strips. I prepared buffers from the Ready Prep 2D starter kit as instructed in the manual. Remove the wet filter paper from the top of the IPG strip. Place the IPG strip into a lane gel side up of the rehydration tray. Add 2 milliliters of equilibration buffer number 1 to each lane of the rehydration tray that you have a strip. Place the rehydration tray onto a shaker walker table for 15 minutes. Note that the rehydration tray has an angled end and a straight end. Whenever you're pouring out liquids, use the straight end so that it does not slide out of the tray. Now add 2 milliliters of buffer number 2. Shake for 15 minutes and pour out. At this point, the strips are ready to run on the second dimension gel. I am going to use BioRad's Criterion precast gels, which accommodate the 11 centimeter IPG strips that we ran earlier. Prepare the gel by removing the sealing tape from the bottom of the gel and slide out the comb from the top. Rinse the wells with good quality DI water. Tap the water out into a beaker and then place the gel sideways and remove with a piece of filter paper the excess water from the wells. Be careful not to touch the bottom of the gel as it can damage the surface of the second dimension gel. We usually do this because unpolymerized acrylamide can cause anomalies in your second dimension gel if it's not removed. To remove the strip from the tray, place the forceps on the tray and gently push on the plastic backing of the gel to push the strip towards the angled edge. Lift up the strip from the right tab. I am going to rinse the strip off in running buffer that I had previously prepared. Dip 2 or 3 times. This will remove equilibration buffer number 1 and 2 that may still be on the surface of the IPG strip. Place the IPG strip gel side up. Place the plastic backing against the back of the cassette. Position the strip centered in the middle of the well with your forceps and gently push on the strip until it touches the gel. And you can tell when it's touching because you'll feel a little bit of springiness, a little bit of resistance. Don't push too hard. Now I will insert a Precision Plus standard plug, which is a broad range standard that has been embedded in agarose, into the reference well on the right side of the gel. Break off one plug, hold onto the handle and break off the spatula, and using the spatula, push out the agarose plug. For Criterion gels, I usually cut the plug into 4 slices to match the size of the well. In this case, I need 1. With forceps, take one of the slices, place on the back of the gel cassette over the reference well, and gently push the plug into the reference well. At this point, we want to seal the IPG strip and standard gel plug into the well of the gel. I will use Protein Plus Overlay Agarose. Heat in the microwave oven until it boils, and when the temperature of the agarose is no longer hot to the touch, take the hot agarose and fill up the well. Do this in one rapid action, side to side. Working quickly, use your forceps to push the IPG strip against the gel to push out any air bubbles. Agarose is going to solidify fairly fast. You can see the agarose filling in the place of the bubbles as they get pushed to the top. Small air bubbles are not an issue. Try to get some of your larger ones, and you should not have any problems for your second dimension gel. Now that the agarose is solidified, I'm ready to run the gel. In this case, I'm going to run the gel in the Criterion gel box, which holds 2 gels. In most cases, you're going to run multiple gels and you want to use a Criterion dodecacell. Remove the gel cassette and place it into the Criterion cell. Load the upper buffer chamber with running buffer. Place the lid onto the Criterion cell. Insert the leads into your power pack power supply. Program for 200 volts for 1 hour. Constant voltage. And start. The second dimension gel has now completed running. You know it is finished when the die front has reached the bottom of the gel. We're now going to remove the gel from the Criterion cassette and prepare the gel for staining. Remove the gel from the gel box, pour out the upper buffer, and open the gel by using forceps or a similar tool by pushing the forceps against the plastic cassette towards you at the top and work your way down. So fold open like so. After I open the gel cassette, I use forceps to remove the IPG strip from the surface of the gel. Starting at the bottom of the gel, gently pull the gel away from the surface of the cassette. The easiest way to do this, turn the gel upside down over the surface of the staining solution, remove a corner, and remove a corner until the gel falls into the solution. Biorad offers many different staining choices for 2D gels, ranging from the visual stains, the traditional Kamasis and silver stains, to the fluorescent stains, Flamingo and Cypro. In this case, I am staining this gel with BioSafe Kamasi, and the first step requires 3 10-minute rinses in water to remove SDS from the gel. We are now going to stain the gel using Biorad's BioSafe Kumasi stain. Following the 3 10-minute water washes, we're going to transfer the gel to a tray of BioSafe Kumasi, and we are going to stain the gel for 45 minutes to 1 hour. Following staining, we are going to transfer the gel from BioSafe Kumasi back into water, and we are going to destain until you get desired background. At that point, the gel will be ready for imaging. Here you can see that I have generated a gel with a clear background and well-resolved spots. I have now completed the entire process for running a 2D gel. I hope that the techniques and tips will help you when you run your next 2D gel. As you have seen, Clean Sample and the Biorad 2D Starter Kit will help you generate good quality, reproducible 2D gels. For additional information and techniques and tips on sample preparation and 2D gel electrophoresis, you can visit our Expression Proteomics website at www.expressionproteomics.com.
So that's it for student for today. I will share these links later with you. Yeah, maybe it's too much for today, all of you. We can do it 9 p.m. the later part. Yeah, then we see us at 9 p.m. today. So I will share these things. So see you then at 9 then. Take care. Bye bye. Thank you very much.